Dear students, in this chapter on Introduction to Food Spoilage by Microorganisms, you will learn about how food gets spoiled and the important microbes involved in the spoilage of food. In this chapter, we deal with the following aspects: introduction, food spoilage and general principles underlining spoilage of food, microbes involved in spoilage of food, major foodborne infections or intoxications. Introduction. Food are not only nutritious to human being, but are also for microorganisms which grow on them. The presence of food poisoning microorganisms and spoilage of food are very important for the consumer in terms of food safety and consumer health. Today, the emphasis is on total quality of food. This means that not only food should be nutritionally balanced, but should be microbiologically safe too. In this chapter, we will understand the general principles of microbial spoilage of food and food poisoning microorganisms. What is food spoilage? We refer to food as spoiled when a food looks bad, which means that there is microbial growth in the form of mycelia or colonies visible on the surface, development of cloudiness in liquids, changes in food color. Food spoilage causes textural changes like slime formation, primarily due to the surface accumulation of microbial cells, tissue softening due to enzymatic degradation, example, soft rot of veggies. Changes in taste and odor like ammonia, amines, sulfides or organic acids. General principles for the spoilage of food. The number and type of microorganisms in food are largely determined by environment from which the food is sourced, microbiological quality of the fruit in its raw or unprocessed state, which is called as intrinsic factor, sanitation during handling and processing, effectiveness of packaging, handling and storage conditions in restricting microbial growth called extrinsic factors. Since food are of plant and animal origin, characteristic of plant and animal tissue that affect the microbial growth need to be understood. The plant and animal have evolved mechanisms of defense against the invasion and proliferation of microorganisms. Hence, fresh fruit and vegetables, if undamaged, can effectively stay fresh without contamination. However, during harvesting, packing and transportation, the skin of the fresh fruit and the vegetables get damaged, thus inviting spoilage organisms. Knowledge about the source of microorganisms and factors influencing microbial growth is essential for food microbiologists. Desirable growth conditions of microorganisms for fermentation and single cell protein production, and finally to understand the undesirable conditions utilized for food preservation. The various sources through which microorganisms gain entry into the food are shown in table 1. Microorganisms in food can be used for preservation like curd or behave as spoilage organisms. A number of intrinsic and extrinsic factors affect the microbial growth. Table 2. Intrinsic are parameters within the food like pH, water activity, oxidation reduction potential, that is EH, nutrient content, antimicrobial constituent and biological structure play a major role in food spoilage. Extrinsic or environmental parameters affects both food as well as microorganism. These parameters include temperature of storage, relative humidity of storage environment and concentration of gases in the environment. Let us learn both the parameters. Intrinsic parameters. Nutrient. Microorganisms need water, energy source, nitrogen, minerals, vitamins and growth factors in order to grow and function normally. Since food are rich source of these compounds, they form a good media or environment for microorganisms to grow. Microbes which cannot use these resources present in the food do not grow in that environment. Among the various groups, molds or fungi can grow in almost all the conditions, as they can utilize most of the nutrient present in food, followed by yeast, gram-negative bacteria and gram-positive bacteria. Many microorganisms present in the food have the ability to utilize simple sugars and amino acids. Few others are able to utilize complex carbohydrates such as starches and cellulose as source of energy. Fat are the least favored source of energy. The common utilized nitrogen source by microorganisms are amino acids. However, almost all microbes utilize proteins, peptides and nucleotides. In general, single compounds are utilized first by the majority of the microorganisms. Water activity, abbreviated as AW. Water is often the major constituent in food. Even relatively dry food like bread and cheese usually contain more than 35% water. However, the water present in the food which is available for the microorganism to thrive on is explained as water activity. Water activity is the partial vapor pressure of water in a substance divided by the standard state of partial vapor pressure of water. In the field of food science, the standard state is almost often defined as the partial vapor pressure of pure water at the same temperature. It can be simplified as the ratio between the vapor pressure of the food when in completely undisturbed balance with the surrounding air and the vapor pressure of pure water under identical condition. Water activity in practice is measured as the equilibrium relative humidity, abbreviated as ERH, and is given by the formula water activity, that is AW is equal to ERH divided by 100. Various organisms thrive at varied water activity. A few are listed in table 3. pH and buffering capacity. The pH or hydrogen ion concentration H plus of natural environment varies from about 0.5 in the most acidic soil to about 10.5 in the most alkaline lakes. The range of pH over which the organism grows is known as the pH range for that organism. This range can be divided into 3 regions: minimum pH, below which the organism cannot grow; maximum pH, above which the organism cannot grow; and the optimum pH, at which the organism grows the best. Microorganisms which grow at an optimum pH well below neutral range, that is 7.0, are called as acidophiles. Those which grow best at neutral pH are called neutrophiles, and those that grow best under alkaline conditions are called alkalophiles. In general, bacteria grow well in the pH range of 6 to 8, yeast 4.5 to 6.5, and filamentous fungi 3.5 to 6.8. However, there are exceptions like lactobacillus and acetic acid bacteria, which have an optimum pH between 5 and 6. Some of the pH requirements of the various organisms are listed in table 4. Redox potential or EH. Microorganisms display varying degree of similarity to oxidation reduction potential of their growth media. The oxidation reduction potential is measure of the tendency of a chemical species to acquire or release electron and thereby be reduced. When an element or compound loses electron, it is called as oxidized, while a substance that gains electron gets reduced. Thus, a substance that readily gives up electrons is a good reducing agent, and while one that readily gains electron is a good oxidizing agent. When electrons are transferred from one compound to another, a potential difference is created between the two compounds and is expressed as millivolt or MV. If a substance is more highly oxidized, the more positive will it be its electric potential and vice versa. The OR potential of a system is expressed as EH. Aerobic organisms require a positive EH value for the growth, while anaerobic organism requires negative EH values or reduced. The redox potential in food is the result of several factors: redox couples present, ratio of the oxidant to reductant, pH, production of toxin, availability of oxygen, and microbial activity. Extrinsic parameters: temperature of storage. Microorganisms have been found growing in virtually all temperatures. A particular microorganism will exhibit a range of temperature over which it can grow in the same manner as pH. Taking the human body temperature as a central point, they are classified as organisms with an optimum temperature near 37 degree are called as mesophiles. Organisms with an optimum temperature between 45 degrees and 70 degrees are thermophiles, example Bacillus, Clostridia, etc. Some archibacteria with an optimum temperature of 80 degrees or higher and a maximum temperature as high as 150 degrees are referred to as extreme thermophiles or hyperthermophiles. The cold loving organisms, which are psychrophiles, defined by their ability to grow below 15 degrees to 0 degree. Psychrotrophs are the main organisms which attack food stored in refrigerators, example Listeria. The maximum damage is caused by mesophiles and psychrotropic organisms in food. Relative humidity of storage environment. Relative humidity and water activity are interrelated. When foods with low water activity are stored in an environment of high humidity, water will transfer from the gas phase to the food and thus increase the water activity of the food, leading to spoilage. Temperature also plays a major role in the modification of relative humidity. The general thumb rule is that at higher temperature, lower is the relative humidity and vice versa. Hence, it would be better to store food in conditions of low relative humidity to increase their shelf life. This can also be carried out by approximately packing the food material. However, variations in storage temperature should be minimum to avoid surface condensation in packed foods. Gaseous atmosphere. Oxygen is one of the most important gases which come in contact with food. This gas influences the redox potential, which in turn affects the microbial growth. This understanding has led to designing modified atmospheric packaging, which will be dealt in Unit 9 or food preservation. Microbial spoilage of food is due to the activity of these organisms. On some of the constituents of the food, they may be broadly be classified into three groups. Fermentation. Carbohydrate in food plus saccharolytic microorganism produces organic acid plus alcohol plus gases. Putrefaction. Protein in food plus proteolytic microorganism produces amino acids, amines, ammonia and hydrogen sulfide. Rancidity. Fatty foods plus lipolytic microorganism produces fatty acids plus glycerol. Microbes involved in spoilage of food. Spoilage defects in food. As microbes are present everywhere, they find ways and means to grow on food. However, as these foods provide a congenial atmosphere for the microbes to grow, they colonize. Such colonized microbes cause spoilage. The spoilage may be by changing the physical appearance of the food or flavor and taste, that is chemical quality. The common spoilage defect that occurs in different food, with some examples are shown in table 5 and 6. The spoilage is not only by degradation of food, but by the synthesis of various products like pigments and polysaccharide, leading to discoloration and formation of slimes. In general, it is considered that fresh fruit and vegetables are infected with fungi. Meat and egg products along with processed foods are spoiled by yeast and bacteria. This is mainly due to the presence of enzymes which break cellulose and pectin, the major constituent in fresh fruit and vegetables, whereas protein degrading enzymes are common in bacteria, hence they spoil milk, meat and fish products. Sugary substances are normally attacked by yeast, as it is in the case of fruit juices. However, there are exceptions like bread, where even though it is a processed food, spoilage is mainly by fungi. Food with high amounts of fat are normally spoiled by bacteria due to their capacity to produce lipolytic enzymes. They mainly belong to the group of Lactobacillus. Major foodborne infections or intoxications. What is foodborne disease? Safe, nutritious food are essential to human health and well-being. However, foodborne disease pose a significant problem worldwide. Foodborne disease is an illness resulting from the consumption of food contaminated with one or more disease-producing agents. These include bacteria, parasites, viruses, fungi and their toxic products. The World Health Organization or WHO estimates that 1.5 billion cases of foodborne illness causes about 3 million deaths every year. More than 250 different foodborne diseases have been described. These different diseases have different symptoms. Hence, indicating specific symptoms to foodborne illness would be difficult. However, the first symptom of food poisoning are nausea, vomiting, abdominal cramps and diarrhea. These are listed in table 7. Foodborne infection. Foodborne infection is caused by the ingestion of food containing live microbes which grow and establish themselves in the human intestinal tract. Some bacteria, virus and parasites cause foodborne illness via infection. The foodborne bacteria that causes infections are Salmonella species, Listeria monocytogenes, Campylobacter jejuni, Vibrio parohemolyticus, Vibrio vulnificus and Yersinia enterocoletica. The most common foodborne parasites are Trichinella spiralensis, Echinus simplex, Gardenia didynalensis, Toxoplasma gondii, Cryptosporidium parvum and Cyclospora species. Foodborne intoxications. Foodborne intoxications is caused by ingesting food containing toxins formed by microbes. An intoxication results when a person eats food containing toxins that causes illness. Toxins are produced by harmful microorganisms, the result of a chemical contamination or a natural part of the plant or seafood. Some bacteria cause intoxication. Viruses and parasites do not cause foodborne intoxication. The foodborne bacteria that causes intoxications are Clostridium botulinum, Staphylococcus aureus, Clostridium perfringens, and Bacillus cereus. Chemicals that can cause intoxication includes cleaning products, sanitizers, pesticides, and metals like lead, copper, brass, zinc, antimony, and cadmium. Seafood toxins include Ciguatera toxin, Cryptobacterioid toxin, shellfish toxin, and systemic fish toxin. Plants and mushrooms can also cause an intoxication. The major food toxin is produced by Clostridium botulinum, known as botulin. It is so toxic that 1 microgram of the toxin can kill 1 lakh people. Hence, in food processing industry, the mere presence of 1 spore in 250 food packs calls for the rejection of the complete batch. Fungal toxins. Where conditions are right, fungi proliferate into colonies and produce toxins known as mycotoxin. The reasons for the production of mycotoxin is not fully understood, as they are not necessary for the growth or the development of the fungi. The production of toxin depends on the surrounding intrinsic and extrinsic environment, and these substances vary greatly in their toxicity depending on the organism infected and its susceptibility, metabolism, and defense mechanisms. Types of fungal toxin. Aflatoxin. Aflatoxins are a type of mycotoxin produced by Aspergillus species of fungi such as Aspergillus flavus and Aspergillus parasiticus. The term aflatoxin refers to 4 different types of mycotoxin produced, which are B1, B2, G1, and G2. Aflatoxin B1 is a potent carcinogen and has been directly correlated to adverse health effects such as liver cancer in many animal species. Aflatoxins are largely associated with commodities produced in the tropics and subtropics such as cotton, groundnut, spices, pistachio, and maize. Ochratoxin is a mycotoxin produced by Penicillium and Aspergillus species. Aspergillus ochraceus is found as a contaminant of a wide range of commodities including beverages such as beer and wine. Aspergillus carbonaceus is the main species found in wine fruit, which releases its toxin during juice making process. Citrinnin is a toxin that was first isolated from Penicillium citrininum, but produced by most of the species of Penicillium and several species of Aspergillus. Some of these species are used to produce human foodstuffs such as cheese, Penicillium camberti, sake, miso, and soya sauce, Aspergillus oryzae. Citrinnin is associated with Alod rice disease in Japan and acts as a nephrotoxin in all animal species tested. Ergot. Alkaloids are compounds produced in the sclerotia of species of claviceps, which are common pathogens of various grass species. Ergotism is a human disease historically known as St. Anthony's fire. Platulin. Platulin is a toxin produced by P. expressum, Aspergillus, Penicillium, and Palsiomyces fungal species. Palsiomyces expressum is especially associated with a range of moldy fruits and vegetables, in particular rotting apples and figs. Although platulin has not been shown to be an carcinogen, it has been reported to damage the immune system in animals. Fusarium toxin are produced by over 50 species of Fusarium, which infect the grain of developing cereals such as wheat and maize. Thus, food is attacked by a number of microorganisms, which indicates that what is good for us is also good for the microorganism. The battle between the man and the microbe will continue in terms of food production, processing, and preservation. Conclusion. In this chapter, we try to understand the commonly occurring food spoilage organisms, the conditions which lead to their growth and proliferation on food determines the extent of spoilage. The conditions may be due to the environmental or extrinsic, or due to the original character of the food or its handling and processing. The causative organism need not be present to call the food spoiled, but the metabolite produced in the form of toxin can cause sickness. Quite often, the food may not look spoiled externally, but may carry these toxins which would cause the consumer to become sick. The sources of contamination may be soil, water, air, human pathogens from manure, that is night soil. It can also come during harvesting, both hand picking and machine picking, which causes high damage if crop is ripe. Contamination may come from processing plant, packaging containers which are reused without any proper sanitation, or during markets due to handling and cross contamination. Thank you, students.